Okay, guys. Welcome back. Today we're going to be going over Unit 4 of the AP Bio curriculum, which is cell communication and the cell cycle. The cell cycle. So, cells, just like everything else in life, there's going to be communication that happens between the cells, and there are different mechanisms that it uses. So in cells they send out chemical signals, uh, which they send out chemical signals, and these signals are like the signals that they send out. The molecules are called ligands, and there are the different methods of there are like different types of cell signal. So there is autocrine signaling, which is the cell basically talking to itself. So auto means self, and then there is juxtacrine, which is direct contact between one cell and another cell. And then there is paracrine signaling, which is between like short distances. So the ligands travel a short distance. And then there is endocrine signaling, which is when the ligands travel a long distance. So let's look at an autocrine signaling, an example. Well, this is a model, not really example, but the cell secretes its own ligand, and then the ligand binds to a receptor on the same cell that secreted the ligand. The root word auto means self, so that's why it's called autocrine. So let's say we have a cell over here. This is our cell. All right, it produces a ligand. This triangle is a ligand. It leaves the cell, so the triangle. Let me use a different color for the triangle actually. It's a big one. Okay, so let's say this is the ligand. All right, whatever. We'll put a key up here. So that's our ligand. It leaves the cell, and then it travels to a receptor on that cell. I'm gonna make it green. All right, and then it binds to that receptor on the same cell, and it triggers a response. Response is triggered, and this is like the cell, it stimulates itself, and it's can be for like cell growth or whatever, but this is common in like cancer cells, where cancer cells, they secrete its own growth hormones, and it will stimulate the cancer cell to keep growing and divide. It's out of control. But this is autocrine signal. Basically, the cell sends out its own ligand, its own signal, and then it binds to itself, and then it triggers its own response. The next one is juxtacrine signaling. So it's direct contact, and it's usually due like a surface receptor on a cell. So we have two cells. All right, they are very close to each other. They're like right next to each other. So this is our first cell, and then our second cell. So cell one is secrete a ligand. So our blue triangle is still going to be our ligand. And then on the surface of cell 2, there's going to be a receptor. And then since they're right next to each other, they're able to bind to each other And then when these two bind, the ligand binds with the receptor on cell 2, it's able to trigger a response for cell 2. And that is juxtacrine signaling. And then paracrine signaling is short distance. So it's usually for neurotransmitters where a cell, they secrete a ligand that travels a short distance and it reaches the receptors on a different cell. So let's say this is cell, so this is like a neurotransmitter, all right? Secretes, so there are these neurotransmitters. Neurotransmitters are ligands and it's being secreted, so it leaves the cell, all right? Leaves the cell. They don't have to be in the same orientation. I don't know why I'm drawing them upwards, but it can be all over the place, but they're the same shape. And this is the like space between cell. I believe it's called the synaptic gap in like neurons. All right, and then here is a target cell. So this is our original cell. I forgot the fancy name for it. Original cell, and then this is our target cell. There are receptors on basically the surface of the cell that accepts the that accepts these ligands, and then once these ligands bind to the receptors, a response is triggered in the target cell. All right, let's just represent that with cellular response. All right, and then now the next one, since we have a long distance, we're gonna need a since we have a short distance, now we need a longer distance, which is endocrine signaling. All right, it's long distance, and it's the ligands. We call the ligands in endocrine signaling, we call them hormones. And hormones are like secreted by like part of your body, and it goes through your bloodstream to reach another place. So the most famous example of a hormone is insulin. So it's for like the blood sugar in your body. So your pancreas, like this, so this is your pancreas, secretes insulin. So insulin, all right, I'm gonna make it a circle. All right, it's secreted, and then the insulin travels into your bloodstream. All right, this is bloodstream, and then it goes to wherever it needs, and it goes to the one of like the body cell that would need it. All right, and then the let's make this a body cell, and then it will bind to a receptor on that cell to trigger a cellular response for that cell. And then it will travel from the bloodstream and it will bind to a receptor. And that is the endocrine signaling. So endocrine is similar to the endocrine system of your human body. So like hormones are sent out from one place of your body and then it travels through the endocrine system into another part of your body and triggering a response for those cells. So signaling, okay. So okay. So cellular, so then now after the ligands are received by the cell must be able to respond to it and trigger a response. This is signal transduction. And it's where the cell responds to the signal, and the process, it can be like gene expression, cell growth, cell division, release of hormones. That is the response that can be triggered from signal transduction. So signal transduction, this is kind of like the general path, all right? So first, signaling molecule, which is ligand, bind to a receptor, then a signal transduction pathway is triggered, and then whatever happens there, it will cause a cellular response. So, signaling molecules. Ooh, before we go on to the signaling transduction, we're gonna have to focus on the different methods that the signaling molecule or the ligands can enter or contact the cell or communicate with the cell. So, if you remember from unit 2, the cell has a cell membrane. So, this is the cell membrane. The cell membrane is a phospholipid bilayer. Phospholipid bilayer. So it has two layers of phospholipids, and so it looks like this. So it looks something like this. So we have a bunch. They're usually closer together, but so they're like this. And then we have a phosphate group, which is the head. Phosphate group, and then we have a lipid tail. So the heads are hydrophilic. Hydrophilics. It loves water, whereas the tails are hydrophobic because they're lipids and hates water. The cell membrane overall is nonpolar, and it's nonpolar, so that means hydrophilic. So that means hydrophobic substances can easily enter through, like, diffuse through the cell. So the easiest molecule to travel through the cell is small and nonpolar molecules. But then other non-polar molecules, like hydrophobic signaling molecules like steroids, lipid hormones, they can diffuse through the cell membrane, and it can bind to a receptor inside the cell. So let's move this here. Gonna move that here. And then this is gonna be, let me move this down here. So it's gonna be, so this is extracellular, which is outside the cell. And then we have intracellular, which is inside the cell. So this would be the environment, and this would be the cytosol or the cytoplasm. So if it's, let's say it's like a small nonpolar, sorry, small nonpolar molecule, it can easily diffuse through the membrane. But then like steroids, lipid-based hormones, they are bigger, they are big but nonpolar uh, molecules, so they are able to diffuse through the membrane, and they bind to a receptor inside the cell, or like inside the cytosol. So it's able to diffuse, and then it can bind to a receptor inside the cell. But what if you have, let's say, a hydrophilic? What if you have a hydrophilic signaling molecule, or if it's a charged signaling molecule? So if you remember from AP Bio Units 2, big, polar, or charged molecules are hard to enter, hard to diffuse due to cell membrane, or so they must go through channels that facilitates the diffusion across the membrane. So uh, there are so then let's draw another phospholipid bilayer cell membrane. Since the some of the molecules they can't go through the cell membrane, they will have to go through special channels. So let's put that right here. So these channels they are specific to whatever it can pass through them. So the membrane receptors it can be like ligand gated ion channels. So the ion channel will open or close whether there is a particular ligand that is binded to that channel. So a famous one is the sodium channel, where it will open or close when a ligand is binded to the binding site, which is over here, and then the channel opens up, and ions are able to diffuse through the channel into the cell. And then there are enzymes on the cell membrane that helps it, and there is G protein link receptor, which is something that is going to be covered. Okay, so this is the first part of signal transduction, which is reception, when the ligand is kind of received by the cell. And now the next part is the transduction. This is the transduction process. So the process behind it is really complicated. It's a series of chemical reactions that is triggered after the ligand binds to its receptor, and then the bunch of chemical processes kind of formulate the cellular response. So there can be signaling cascades for the response. If I can see a picture. So let's say we have the cell membrane over here, and then we have a ligand which is binded to the stingy to the receptor. It's gonna cause a reaction, right? And then it's going to cause another reaction, and then that causes another reaction, and that is our final response. All right. But then if we have like a hydrophobic process, a hydrophobic ligand, that's a little bit more straightforward. So the signaling molecule is able to just enter the cell through the fusion, and then it can even diffuse through like the membrane of the nucleus, so it can enter the nucleus and activate. So let's pretend that this is the nucleus, all right? And then it can just enter through the nucleus through the fusion again and bind to a receptor in the nucleus and trigger gene expression. So that is for hormones. And then there are proteins that are involved uh, in this process. So there are many steps, and it's very complicated. Many signaling transduction pathways include a series of protein kinase that adds a phosphate group to the next protein in the cascade sequence. So I did this example in class where a cellular response is triggered, and then ATP is floating around the cell, and then kinase removes a phosphate group from the ATP, and then it gives a phosphate group to the next kinase, and then that kinase repeats the process, and then finally it leads to like a response given by the cell. So kinase is important, which adds so the purpose of kinase ASE, that's the suffix, it's an enzyme, and it adds a phosphate group to the next protein in the cascade sequence. And yeah, bacteria cells, their signaling whatever is a lot more simpler. Um, there's also phosphatase, which are also an enzyme, but instead of adding, it removes a phosphate group. And then enzymes, they all produce, well, their goal is to produce uh, secondary messengers, which like the example is cyclicase, which produces cyclic AMP or CAMP from ATP. And then that it triggers further cellular responses. Right, and then now the response will be the final step of the signal transduction, and it's like the kind of the result, the ultimate goal. Examples of cellular responses can be like activation of genes, it can be opening ligand gate ion channels, initiation of cell processes, and like the cause, like the initiation of apoptosis, which is programmed cell death. If I miss anything. All right. So, feedback mechanisms. So, cells, if you recall from like just general high school biology, if you remember or if you have taken it before, cells, your human body or basically like an organism is always trying to maintain homeostasis, which is just the maintenance of a stable state. So there's going to be feedback that kind of regulate the processes. So there is first, there are two. There is negative feedback pathway. All right, negative, remember the word negative, or it's also called feedback inhibition, which works by turning itself off using the end product of the pathway. So essentially, if enough X is made, then the pathway to make X can be turned off. So that is for like production, but then in your body for like more homeostasis. Let's say the body temperature, your body temperature gets too high, then cell signaling, it signals skin cells to release sweat to lower down the temperature. And then once the body temperature is lowered to a certain range, the cell would send signals to stop stop the cells from releasing more sweat so that you stay at the range of temperatures. And then ATP can be, well, this is like another really famous example or like important one that you need to know, which um is the control of blood glucose levels. So blood glucose, if you remember, there's insulin, but there's also glucagon. So after you eat something like sugary, all right, your blood sugar levels will rise, and your pancreas will try to release insulin, which is a ligand, which is also a hormone. Ligands, so hormones are ligands, but not all ligands are hormones, right? And then so insulin is the hormone and ligand in this negative feedback system. So the insulin will cause the body cells to absorb the glucose from the blood so that the glucose level can return to normal range. And that is negative feedback because your glucose level rises, it goes high. So then your body wants to lower it, so it triggers a response to lower the blood sugar level. And then conversely, if your blood sugar gets too low, your pancreas will release glucagon, which stimulates the liver cells to break down glucogen into glucose. So your body forms glucogen from glucose for like digestion and then just storing it. But then when your blood sugar gets too low, glucagon is released into your body so that it can signal the liver to break down glucogen into glucose so that your glucose level of your blood is back in the normal range. And then now positive feedback is slightly different. All right, well, it's very different, but it basically magnifies the process. It further stimulates a process. So once X is made, then it tells the pathway to make even more X. So this is less common than negative feedback pathway because you usually say, because negative pathway is like, oh, there's enough of this, then we should stop making it. But then X, for positive feedback, it's like, oh, this is made, so we should make more of this. So positive feedback is usually used for fruit ripening and labor. In labor, there's a hormone called oxytocin. I don't know how to say it, but it stimulates the contraction of the uterine muscles during labor, and the contraction of the muscles trigger the production of even more oxytocin, which triggers more contraction of uterine muscle, which is this would be a positive feedback. So here's like a little diagram. So for negative feedback, when the stimulus, all right, when there's more stimulus, so the stimulus increases, then the response would decrease. And then for positive, when the stimulus increases, the response also increases. So for negative feedback, when there's more stimulus, so like more insulin is produced, then the response would be that the blood sugar is lowered. But then for positive feedback, more oxytocin is produced, and as a result of that, even more oxytocin is produced. So that is positive feedback. And then yeah, in plants, plants use electric signals to respond to like environmental stimuli. So it can be for like flowering, respiration, photosynthesis, wound healing, and there are light receptors in plants that kind of tell you like the timing of flower and chlorophyll production. Some plants can even use chemicals to communicate with nearby plants. So, for example, wounded tomatoes produce a volatile chemical signal as an alarm signal for nearby cells. So nearby cells can like prepare for a defense or an immune response. And that's the part with that's basically it with cell signaling. And then after cell signaling is the cell cycle, which I'm going to do in a separate video.